Okay, it's 12 o'clock, we're live. So today's first speaker is Professor David Schatz, who's Professor of Immunobiology and MD&B. And his title today is What Prevents Catastrophic Instability of the Lymphocyte Genome During Antigen Receptor Gene Assembly.
All right. All right, good morning or good afternoon or whatever it is. And I would be more than happy to entertain questions, obviously at the end of the talk, but also during the talk. So if you have questions, thoughts, suggestions, anything, please just jump right in. Let me know. There's plenty of seats, no reason for people to be standing in the back or anyway. So anyway, please interrupt with questions. So as Dave said, I'm in the Department of Immunobiology. I've been here for a couple decades now and have been for most of my career studying questions relating to the assembly of the antigen receptor genes and their diversification. So let me just put this in a biological context for those of you who don't think about lymphocyte development very often. Almost everything that I study is somehow involved in B cell development and antibody gene assembly and diversification. So the process of B cell development is a series of highly orchestrated developmental transitions that are organized around the rearrangement and assembly of the antigen receptor genes, the immunoglobulin genes. So first the heavy chain gene rearranges in the pro-B cell, the light chain gene rearranges in a pre-B cell, and successful rearrangement allows the cell to express an immunoglobulin on its surface, and it becomes an immature B cell, which then is exported to the periphery where it will reside in lymph nodes, spleen, and is called a mature B cell, but a naive B cell. And if that B cell is fortunate enough to interact with antigen and receive appropriate T cell help, it will be activated to engage in the immune response, and that process leads to further diversification of the antibody genes through processes known as somatic hypermutation and class switch recombination, which again are taking place in germinal centers in lymph node and spleen. And my lab studies both these early gene rearrangement processes and these later gene diversification processes. And most of what we'll focus on today is the early steps, or V DJ recombination, which is involved in putting the genes together. These immunoglobulin genes are highly unusual in that they pass through the germline in a disassembled configuration. They have to be put together. This is the heavy chain locus. It's assembled in two steps. First, the D gene segment is joined to J, followed by V joining to DJ. And then, and only then, can you express a productive heavy chain. And then a similar set of processes, V to J, is rearranging the light chain locus, and then you can make an antibody. And this part of the antigen receptor gene is what's encoding the part of the antibody that's actually involved in antigen recognition. So after V DJ recombination puts the heavy chain together, it can undergo further diversification, and I should say V DJ recombination is mediated by RAG1 and RAG2, which I'll say much more about in a moment. But after that process, the further diversification is mediated by a completely different enzymatic system, and that's driven by the activation-induced deaminase. And I won't have time to talk about AID at all after the next slide. But just to put it on your radar screen, AID mediates processes known as immunoglobulin gene conversion, somatic hypermutation, which puts point mutations into the variable region gene segments, and it also is the key enzyme triggering class switch recombination, which allows you to go from, for example, expressing the IgM or mu isotype of the heavy chain, for example, to epsilon, and this would be now making an IgE antibody. And the main reason that I wanted to introduce both of these processes today is that there is an incredibly strong link between RAG and genome instability and between AID and genome instability, and they are major players in the genome instability that is seen in lymphoid malignancies. And this is just some of the examples. I'm obviously not going to go through all of the individual examples, but RAG and V DJ recombination have been implicated in a whole bunch of different kinds of deletions and chromosomal translocations and other instability events found predominantly in pre-B or pro-B or early lymphocyte malignancies of developing B cells, whereas AID and the processes of class switch recombination and somatic hypermutation are major players in instability found in mature B cell lymphomas, especially DLBCL, but also Burkitt lymphoma, where they trigger particular MYC-IGH chromosomal translocations. I want to point out that RAG is expressed in both developing B and T cells, so you'll see it involved not just in B cell, pre-B cell lymphomas, but pre-T cell lymphomas as well, because it's also rearranging and assembling the T cell receptor genes. AID, however, is restricted to the B cell lineage, and so it's specifically involved in lymphomas and leukemias of B cells. And then there's even some suggestion that RAG and AID in some unusual events could collaborate in these so-called CPG type translocations in certain kinds of lymphomas to create genome instability. So these are major players in instability, and one of the major focuses of work in my lab is to understand what targets RAG and AID appropriately and why are they sometimes mistargeted, creating this kind of havoc. So let's now zoom in on V DJ recombination and think about how this reaction is taking place. So now imagine this is one of the gene segments, a V or a D or a J, and this blue rectangle over here is another one, and they're going to undergo recombination. The major players are the RAG1 and RAG2 proteins and an accessory factor I'm not going to say more about. These proteins get together and bind to the DNA adjacent to one of the two gene segments, and they bind to this triangle here, which is a recombination signal sequence, which I'll say more about in a moment. This is the sequence-specific binding site for the RAG proteins. They bind to one recombination signal, and then they're going to capture the second one in a synaptic or paired complex. Now, these recombination signals can be a few hundred base pairs apart, or they can be megabases apart. So these reactions can span huge genomic distances. They form the synaptic complex, and it's within this complex that RAG is activated as an endonuclease to cut the DNA, and it does so in a quite distinctive manner. I'm not going to go into the details, but it makes a break in the DNA immediately adjacent or between the recombination signal and the gene segment. And at this point, RAG is pretty much done. It's going to hand things over in a very orderly and actually very importantly orchestrated fashion to a whole group of DNA repair factors of the non-homologous end joining pathway that then process these ends and ligate them together. Typically, the piece of DNA containing the recombination signals is deleted as a circle and is eventually lost from the cell, whereas the coding segments, the V's and the D's and the J's, are joined together in an imprecise manner, giving you this what's called this coding joint here. So that's the overview of the reaction. And obviously, the danger zone is right here. When the DNA has been cut, if these ends are not joined properly, you can end up with all kinds of bad things where these ends can participate in deletions, translocations, duplications, and other bad things. So let me give you a little bit of an overview of the RAG proteins. RAG1 is the major player. It has, each of these proteins has a core region, which is the minimal essential region required for activity, and the RAG1 core sort of does it all. It has the DNA binding domains, it has the active site for DNA cleavage. These acidic residues here are critical for that. And so it seems like it should be able to do everything. Oh, it also has this big, poorly understood N-terminal region that has ubiquitin ligase activity, is almost certainly very important, but we don't understand it very well. But this region here is the major player in the reaction with all the DNA binding and cutting activities, and yet, if you don't have RAG2, this reaction just doesn't go at any appreciable efficiency. So RAG2 is sort of an accessory factor enhancing the DNA binding, and it's critical for DNA cleavage activity by RAG1. But it does have, and we actually understand very little about the function or structure of the core of RAG1, of RAG2. But RAG2 has a very interesting PhD finger in its sort of non-essential C-terminal region, which has the very interesting property of binding to a particular chromatin or histone modification, histone 3 when lysine 4 is trimethylated. This is a marker of active chromatin. I'll come back to it in a moment to explain more about the significance of this. But this marks active or potentially active promoters particularly. It's also found in a few other places in the genome. But any active gene will be marked with H3K4 trimethyl, and this PhD finger of RAG2 binds quite avidly to this mark, as you will see in a moment. So about 10 years ago, we set out to understand how this reaction is regulated, and our focus was beginning to just ask, hey, where in these large, complex antigen receptor loci, immunoglobulins and T-cell receptors, where do the RAG proteins bind to initiate V DJ recombination? And the answer, which was a lot of heartache and a lot of hard work through the work of Yanhong Ji, a postdoc in the lab, she finally got the chip, the chromatin immunoprecipitation working for the RAG proteins, and she was able to define that the binding of the RAG proteins in these loci happens in very focal, specific regions right over the J gene segments. And so these loci, which can be megabases in size, have RAG binding domains of only a couple kb, 2, 3, 5 kb, right over the J gene segments. This is very appealing to us because it meant that if you want to regulate V DJ recombination, you could focus your efforts on these places here where the RAG proteins are bound. And so these are what we call recombination centers, these small regions of very active chromatin within which the reaction is orchestrated. All right, so that's great. So that's V DJ recombination. But what about genome instability? Because this is where it's supposed to happen. But what about the rest of the genome? So that led to this question. Where else in the genome do they bind? If they do bind elsewhere, why do they do so, and what's driving that? And this has been a close collaboration with Rafael Casellas' group and work that's been driven by a fabulous postdoc in the lab, Grace Teng. And the method that we've been using is to do chromatin immunoprecipitation followed by high-throughput next-gen sequencing, ChIP-seq, using some monoclonal antibodies we've developed fairly recently to the RAG proteins. Okay, and so the answer was actually fairly shocking to me, that RAG1 and RAG2 bind to thousands of sites in the genome of developing lymphocytes. And this is just showing you a fairly random piece, about 600 this is the genome browser showing you different tracks of ChIP-seq data. This is about a 600 kb piece of chromosome 11. You can find many, many, many other places in the genome that look very similar. And let me just take you through this so you can get oriented. Down here you can see the genes are annotated, and this orange track is the H3K4 trimethyl. Remember, that's a marker for active promoters, and you can see that this is data from the mouse thymus, and you can see that there's a lot of active promoters in this region, which is why we picked it. TCF7 is a particularly important and highly expressed gene in mouse thymus, and there's H3K4 trimethyl marking those active promoters. If you look at RAG2, which is in green, you can see it's an almost perfect mimic of H3K4 trimethyl. So it is being pulled into these active promoters, presumably by interaction using that PhD finger to bind to this histone mark. And I defy you to be able to distinguish between this track and this track. It's almost perfectly identical. And this is the control. This is the RAG2 ChIP-seq with a RAG2 knockout thymus. And you can see it's basically blank. The blue is RAG1, and this was the real shock for us, because RAG1, remember, is the really dangerous enzyme. It's the one that can actually cut the DNA, and it's binding to thousands of sites in the genome. Some of them, like this, are very, very strong binding sites. This amount of RAG1 binding here is the same amount, approximately, that we would see at the antigen receptor loci, where real V DJ recombination is taking place. And yet the signal here is just huge. And then there's lots of other signals. These are pretty weak, and there's ones that are in between that are medium. And everywhere that you find RAG1 binding, you will invariably find RAG2 and H3K4 trimethyl. And the result of that is that the combined binding of RAG1 and RAG2 at these thousands of sites is predominantly about 80% at active transcription start sites. And just to make it clear, we've also done this in mouse pre-B cells. This is the T cell lineage, but pre-B cells show the same thing. And we've also done human thymus, and we know that this is generalizable there as well. All right, so now this is a real puzzle. We have thousands of binding sites of RAG in the genome. Why? What would be the conceivable purpose of having RAG binding all these places in the genome when, as far as we know, its only function is in mediating V DJ recombination at 6 or 7 loci in the whole genome? So we've looked hard for some possible function. We could imagine that RAG binding would influence chromatin structure, epigenetic modifications, gene expression. We've tried to look at all of these, and thus far we've seen no obvious effect on chromatin structure, although we can't rule that out. We haven't seen alterations in epigenetic modifications as a result of RAG binding, nor have we been able to detect any effect on gene expression simply by the presence or absence of the RAG proteins in terms of binding. So thus far, we've basically struck out trying to find a physiological purpose. Some collaborators have some very intriguing data that may be hinting at a purpose, and I'm not going to go into that. Stay tuned, but thus far, we don't see any obvious function. So if there's not a physiological function, are there pathological consequences of widespread RAG binding in the genome? Well, you can probably already guess the answer to this, we think, is yes. But let's look and think about this. Why would we suspect that there might be pathologic consequences? The reason is the following. The recombination signal sequence that RAG1 and RAG2 bind looks like this. This is the consensus recombination signal. A 7-base heptamer motif, a 9-base nonamer motif, that are separated by a spacer of either 12 or 23 base pairs. The details don't really matter. There's only a couple things you need to know about recombination signals. First of all, they come in these two flavors, either 12 or 23 base pair spacers. Recombination only takes place between a 12 and a 23. And then the other thing that's really intriguing is that the recombination signal sequence actually can vary tremendously from this consensus that I'm showing you. In fact, there are only 3 residues of the RSS that are perfectly conserved, and that's the CAC of the heptamer. There are 5 bases of the nonamer that are highly conserved but not perfectly conserved. And so these guys here, many of these positions can vary, and you still have measurable binding and cleavage activity of the RAG proteins. Because of the degeneracy allowed in the recombination signal sequence, there are literally millions of what we call cryptic recombination signals littering the genome, like 1 every kb or more. And so now you see the problem. We have thousands of binding sites for this nuclease, millions of potential cleavage sites, and so we were just wondering, what the heck is it that prevents just meltdown of the genome every time a lymphocyte starts to develop? Well, we don't have a complete answer to that, and I think the answer is going to take us in all kinds of interesting directions. But let me just show you some glimpses of what's going on in the genome. So there are, as I told you, there are some areas that bind huge amounts of RAG1 and RAG2, and ironically, some of the strongest binding sites are the RAG1 and the RAG2 promoters. So RAG1 and RAG2 are adjacent genes sitting here like this, and one of the strongest sites in the whole genome, this is RAG1 binding, this is RAG2 binding, right at the promoter of RAG1. This is H3K4 trimethyl. And computationally, we were able to find cryptic recombination signals sitting here flanking the promoter of the RAG1 gene. And so we expected that there would be nice recombination between these because of the strong binding of RAG that we saw here. And there's another locus, the ETS2 locus. Strong binding of RAG1 and RAG2, and cryptic recombination signal sequences nearby. But when we actually looked for recombination by PCR, we were unable to detect it with either of these pairs of cryptic RSSs. So that got us wondering, well, maybe our computational predictions aren't very good and we're just not good at picking out RSSs. So we then took these RSSs out and did some tests in transient transfection in cell culture to just see if they were active. And actually, we weren't very good at picking out good RSSs. These guys actually did not have detectable recombination activity, and only this one supported some activity and this one didn't. So all right, you can say, all right, so there's no recombination going on here because we just didn't pick out good RSSs. So as I said, one or both of these cryptic RSSs support very little recombination activity. But there are places in the genome where bad stuff is happening. And one of them is the mouse Notch1 locus, which is actually very frequently altered by V DJ recombination in murine T-ALLs. And here's the promoter, and there's some real puzzles here because the amount of RAG binding is very low relative to the two things I just showed you. And here's these cryptic recombination signal sequences that had been identified by other people and shown to undergo recombination in the normal thymus. And Grace was able to confirm that this is a PCR assay looking for this recombination event. And you can see that in some of the assays, Grace has a hit and leads to a frequency of about one event every few 100,000 thymocytes. In a normal thymus is deleting this region. And that's a bad deletion because it screws up Notch1 and can lead to a dominant active form of the Notch1 protein. And just for the aficionados who maybe think about ATM, a very important DNA repair and damage response protein, in the ATM knockout, the frequency of these deletions actually goes up a little bit. But I'm not going to dwell on that. I'll be happy to address it in questions. When we tested these RSSs for activity in our transient transfection assay, interestingly, one was active and the other wasn't. So it just shows you that in vivo, there can be events that we can't detect very well in our simple transient assays. An even more striking example is the mouse BCL11B locus. It has pretty good binding of RAG1 and RAG2 at its promoter, which is right up here. And it has these cryptic RSSs that are 65 kb apart. One of them way down here in a region where there isn't a whole lot of RAG binding. And these cryptic RSSs in the literature were shown to mediate recombination at a frequency about 10 times higher than Notch1. And Grace was able to recapitulate that in our assay. And as before, the frequency of these events went up even higher in the ATM knockout thymus. So these cryptic RSSs are mediating deletions in around 1 to 20,000, 1 to 50,000 cells. And that's just in the normal thymus. And since you have output of millions and millions of thymocytes a day, that means there's lots of cells containing this deletion mediated by RAG. And we measured the recombination activity of these guys, and they're both active. So these are pretty good RSSs that for some reason are sitting there continuing to be targets of RAG-mediated activity. So a sort of intermediate stopping point here in terms of conclusions, and I'll wrap up pretty quickly now. But one of the things that is clear is from the analysis of the RAG and the ETS locus is that poor RSS quality might help to suppress the off-target effects of RAG. Nonetheless, RAG catalyzes deleterious recombination events at some places in the genome, like Notch and BCL11B. And one of the really striking examples of this deleterious recombination was published recently in a paper in Nature Genetics on a particular kind of pre-B ALL. And it's an ALL that has an ETV6 RUNX1 translocation. And this paper from Peter Campbell's group showed that these cells, which express RAG, that contain recurrent deletions, so different tumors will have almost to the nucleotide the same deletion events. They have nucleotides inserted at the junctions, which is a hallmark of VDJ recombination. And strikingly, the deletion endpoints map to promoters of active genes. Remember, that's where RAG binds. And they have a heptamer-like sequence nearby very frequently. So this is just a smoking gun for RAG-mediated recombination that's contributing to the development of this particular leukemia. So this is a dangerous business, this V DJ recombination, and we started to wonder, evolution has to be dealing with this for hundreds of millions of years, because RAG has been around since the origin of the jawed vertebrates. So it's in everything from sharks to humans. And so the question was, has evolution found a way to minimize this risk of genome destruction? And our hypothesis was that the genome has itself evolved to reduce the incidence of cryptic RSSs where RAG was binding. And so that was something we could actually test computationally using what's known as the RSS information or RIC algorithm to identify cryptic RSSs in the genome. And so what we did was we compared the density of cryptic RSSs around transcription sites where RAG binds and compared to those where RAG doesn't bind. And indeed, the density of cryptic RSSs was lower here than here. And the most striking example of that is in these plots here, where genes have been binned into 200 gene bins from the strongest RAG binding down to the weakest RAG binding, and then the density of cryptic RSSs per base pair plotted on the y-axis. And you can see this amazing, almost linear correlation that as RAG binding gets weaker, the density of the cryptic RSSs gets stronger, and that's both true for both cryptic 12s and cryptic 23s. And so we would conclude that RAG, or at least the threat of RAG, has influenced the sequence of the genome during vertebrate evolution in probably pretty profound ways. So what happens if you undo what evolution has done? What if you just go back in and now you insert RSSs back into the genome? Just proof of principle. Can you just put an RSS or a pair of RSSs anywhere you want in the genome and have them undergo recombination? So that was something we could actually do, and the experimental system we used is Abelson transformed pre-B cells, which you don't need to know the details of, but the ones we're using are RAG-inducible. They're ATM deficient, which is important if you want to detect translocations. Otherwise, it's not particularly relevant, and that's totally irrelevant. And so the strategy is we use a tailin, which is a tail nuclease, to make a site-specific cut in the genome. We have a targeting vector, which has its two homology arms, one of which has been subtly modified to contain recombination signals. So you knock this into the genome of the pre-B cell, you knock in the RSSs, and then you remove the selection cassette with Cre recombinase, and so you end up with a pretty much normal locus. Just a few, relatively few number of base pairs have been changed, creating good 12 and 23 RSS and the LOX P site, which is nearby. So those are the only changes to the genome sequence. And the place that we've done that, Grace has done this in several places in the genome. I'm just going to show you data on one of them. This happens to be the AID locus for reasons that were just technical. AID has an enhancer called E1, which is right up here, and what Grace did was insert cryptic RSS, good RSSs at this E1 enhancer right here. The reason this data is important is it shows you that this is a part of the genome that's pretty quiet. It doesn't bind a lot of RAG. It certainly doesn't have a lot of H3K4 trimethyl. And so there's just not much going on here. So we put the RSSs in here, so do they recombine? And the answer is yes, they recombine like crazy. When we put these RSSs into the AID locus, we use a simple PCR assay, we induce RAG, and we look for this deletion event that would be mediated by RAG, which would form this coding joint. And the answer is, this is the parental cells of the uninduced cells, and then after RAG induction, this is the recombined band. And this actually represents something like 10 to 40% of the alleles undergoing recombination. So yes, they recombine like crazy, and we've obtained similar results for RSSs inserted actually into two other places in the genome. And then the final question, do they participate in chromosomal translocations? So we looked, the simple way to do this is to allow, the RAG is making breaks at these RSSs that we've inserted. It's also in these same cells making breaks at the immunoglobulin kappa locus. So can we detect translocations, say, between this end and any of these ends? And so there's a whole bunch of possible translocations. You could see red joined to purple or green joined to purple. You could see the red or green joined to the blue side of things, and then there's other possibilities as well that aren't so important. And the answer is yes, you can detect translocations easily. And that's either of these types of events up here. We can detect translocations only after RAG is induced. They've been sequenced and verified and all the rest. And so the final message that I'm going to leave you with is RAG binds widely in the genome of developing lymphocytes, primarily at these active transcription start sites. It represents a significant threat to genome stability. Evolution has responded to that threat, we would argue, by essentially purifying or trying to purify the genome of threatening or dangerous cryptic recombination signal sequences, particularly in the regions of strong RAG binding. And for us, sort of from an evolutionary point of view, it's interesting to think that maybe, I don't know of many examples of where cancer has been a driving force for evolution, but this might be one of the examples. And finally, we can reverse this. If we put RSSs back in, we can reveal the severe threat that RAG represents. And so with that, I will just thank the amazing work from Grace Teng, our collaborators, Rafael Casellas in particular, and other folks in the lab, and Eric Meffray, who helped with the human thymus, and I'd be happy to take questions. Thanks.